Hello everyone, welcome to Care Microbiology classes. And today the topic is bacteria. Okay, in the 2012 and 2013 question papers, which type of questions they asked about bacteria, different kinds of bacteria, and causing diseases, or bacterial growth and bacterial growth cultures, like that. So let's go to the topic. So I'm starting with the 2012 question paper, okay? And the first question in this question paper is nodules of leguminous plants are a good source for the isolation of bacteria that are capable of. Here a straight question it is, and you know very well the answer also because you faced this question in your intermediate and you also studied I think in 10th also, right? So nodules of leguminous plants are a good source for the isolation of bacteria that is capable of. Leguminous plants nodules contains obviously nitrogen fixing bacteria, right? And the right answer is option A. So the next question is addition of casein to solid media and picking up bacterial colonies that form clear zone is termed as. Here the question what he asked means, so we are adding a casein to the solid media to pick up the bacterial colonies that forms the clear zone, right? Here the question is like that only. You have to understand the question. Like we are adding the casein, right? To pick up the bacterial colony which can form the clear zone means which can proteolyze that casein. We have to pick up that bacteria. So what type of process it is he is asking, right? Differential enrichment or streaking or serial dilution or selective enrichment. Here the answer is differential enrichment. Why it is differential enrichment means we are differentiating the casein proteolizing bacteria from the general bacteria. So the answer is the differential enrichment bacteria. Because why it is not selective enrichment means we are not at all adding any special component that can suppress the growth of other bacteria. We are adding only the substance which can helpful to find out the particular bacteria. That's why it is the differential enrichment. The next question is leishmaniasis is transmitted by option A sandfly, option B TSC fly, option C rodent fly, option D mosquitoes. The correct answer is sandfly, option A. Okay, and see about the leishmaniasis also. Leishmaniasis disease is caused by the Leishmania donovani and it is a type of protozoa and the vector is female sandfly. It is also called as the phlebotomus and it causes the disease visceral leishmaniasis which is also called as the kala azar. The next question, in gram staining of gram negative bacteria the crystal violet iodine complex formed will be washed away after addition of which substance they are asking. See here the options. Option A saffron in solution, option B ethyl acetate, option C water, option D alcohol. Look students, uh, some definite questions will be there in every year like that gram staining and some kind of acid fast staining, gram staining, these are the very common and important topics. So you must have to remember the process of the gram staining I gave here. Please look it. The step one, we will fix the smear by heat fixing process. Okay. And step two, we will pour crystal violet solution. We will wait for 1 minute. Later, we will wash under the tap water and we will pour grams iodine and we will wait for 1 minute and we will remove it and we will put alcohol or acetone and we will wait for 5 to 10 seconds. Later, we will put saffron in and we will wait for 30 to 60 seconds, right? So here what you have to understand in gram staining of the gram negative bacteria the crystal violet iodine complex formed will be washed away after addition of, the answer is alcohol, right? This crystal violet iodine complex is removed using the alcohol, or we may call it as the decolorizing agent, right? So correct answer is option D alcohol. See here the next question. Match the pathogens listed in column 1 with the disease listed in the column 2. Rotavirus, Leishmania, Salmonella typhimurium, Epstein-Barr virus. Right? Here Rotavirus causes the diarrhea. Leishmania causes, already we discussed in the previous question, right? Leishmania causes the Kala Azar. Salmonella typhimurium causes food poisoning. Epstein-Barr virus causes the glandular fever. Here, some tricky, like a confusing thing is nothing but the Salmonella typhimurium and typhus fever. Like the names are similar, right? You may get confused, but remember this typhus fever is caused by the rickettsiosis, okay? And Salmonella typhimurium is always connected to the food related disease. Salmonella typhi causes the typhoid. Of course, fever will be there, but mostly it is related to the food poisoning, okay? Like because of the contaminated food, we will get that fever. Let's move. So, here the correct option is the option B, okay? Let's move to the next question. So obviously, I started the 2013 question paper. Okay. See here the question. The time required for an equally to divide in is less than the time required to replicate its chromosome. This is possible because. So you have to read this question again. The time required for an equally to divide is less than the time required to replicate its chromosome. Means so for the replication of the whole cell, what is the time taken for the replication of whole cell? It is more than the replication of the, sorry, it is less than the replication of its chromosome. Means chromosome, for the replication of the chromosome it will take much time, but the replication of the E. coli, it takes very less time. How it is possible? They are asking the question. See here the options. Option A, of an abundance enzymes required for replication, it contains or it linearizes the chromosome and replicates from both ends. Option C, only one daughter cell receives the nuclear material. Option D, multiple replication forks will be there, right? See here this E. coli contains a circular DNA, right? So see the explanation here. New rounds of replication starts before the previous round is completed. So this creates the multiple replication forks on the same chromosome. So as it is a linear DNA, it cannot, sorry, it is a circular DNA, right? So it cannot be linearizes. So option B is wrong. And abundant enzymes required for the replication, of course it has, but the question is not related to the option A. And the option C, only one daughter cell receives the nuclear material, it is also wrong. And option D, as because multiple replications forks will be there. So before formation of the cell only, because of the multiple replication forks at different areas. Okay. So this nucleic material will starts the replication process, and whenever the cell get replicates, the new DNA strands will also enters into the new cells. That's why the correct answer is the multiple replication fork. I hope you understood what I told. The next question is, a bacterial population contains a mixture of wild type and leucine auxotrophs. From this mixture, the leucine auxotrophs can be enriched by growing the mixture in minimal medium supplemented with. Once again, I'm reading the question. Listen carefully. A bacterial population contains a mixture of wild type and leucine auxotrophs. From this mixture, the leucine auxotrophs can be enriched by growing the mixture in minimal medium supplemented with. So, we have to grow both the wild type and leucine auxotroph. Okay. Where we have to grow it? We have to grow it in the mixture that leucine auxotrophs can be enriched by growing the mixture in minimal medium supplemented with. So in a minimal medium, along with all the microorganisms, wild type and leucine auxotrophs both has to grow. Okay, but they have to grow. Which kind of substance we have to give to that minimal medium? Here they mentioned four options. Option A leucine, option B penicillin, option C leucine and penicillin, option D chloramphenicol. Here, as it is a leucine auxotroph, definitely we have to give the leucine, right? Definitely we have to give the leucine. And what they asked, they asked that the leucine auxotrophs can be enriched by growing the mixture, and we have to grow the wild type also, right? For this leucine and penicillin, we have to control the wild type organisms, and we have to encourage the leucine oxotroph. For that, we have to supplement that media with leucine, and we have to add penicillin also to control the growth of the wild type bacteria. So option C is the correct answer. The next question is, gram positive bacteria are more rigid than the gram negative bacteria because of the option A, presence of multiple layers of peptidoglycan. Option B, lack of D amino acids in their cell wall. Option C, presence of N-acetyl glucosamine in the peptidoglycan backbone. Option D, presence of lipopolysaccharides. Here the correct answer is option A, presence of multiple layers of peptidoglycan. Because if you see that N-acetyl glucosamine is it is not alone present in the peptidoglycan backbone, because NAG and NAM both will be there in the peptidoglycan, right? So, presence of N-acetyl glucosamine is not at all correct answer. And option D, presence of lipopolysaccharides. If you see that these lipopolysaccharides are seen in the gram-negative bacteria, right? And lack of D amino acids in their cell wall is also not a correct answer. Option A is the right answer. Next question. Heliobacter pylori can survive in the highly acidic environment of human stomach because option A, it creates an alkaline microenvironment around itself by urease action. Option B, rapidly invades the stomach cells and escapes the acidic environment. Option C, it capsulated and hence it is protected from acidic stress. Option D, it undergoes sporulation for self protection. Here the correct answer is option A. It produces urease, and because of that urease action it creates alkaline environment and it can withstand the acidic environment in the human stomach. Helicobacter pylori. So it for this you have to see the three other options also. It rapidly invades the stomach cells and escapes the acidic environment. It is wrong because this Helicobacter pylori mainly it present in the mucus layer only. Okay, and option C is also wrong because it is not capsulated, and option D is also wrong because it don't forms any spores. So option A is the right answer. And if you see the next question, the waxy surface of the dental plaques does not allow oxygen to diffuse readily. A sample of dental plaque was inoculated into a rich medium and incubated at 37 degrees centigrade with vigorous shaking. However, there was no bacterial growth. The absence of growth was attributed to the following reasons. Question was little bit very long, right? So you have to read it once, twice also. Read it very carefully. Answer is there only in the question only. So if you read some carefully, you will get the answer. Once again, I will read the question. The waxy surface of dental plaques does not allow oxygen to diffuse readily. A sample of dental plaque was inoculated into a rich medium and incubated at 37 degrees centigrade with vigorous shaking. However, there was no bacterial growth. The absence of growth was attributed to the following reasons. They gave three reasons, right? Bacteria are obligate anaerobes. These bacteria require highly alkaline medium for growth. This bacteria requires a very hard surface, such as that of teeth, to grow. Here, the correct answer is P and R. So, why we will discuss little bit. So, you know that bacteria which present in the plaques, they are obligate anaerobes. But here, what we did, we vigorously shake it that media, culture media, right? After inoculation, we vigorously shake that culture media. So, when we are shaking that culture media, it will get oxygen. So, no bacteria will be alive. And next thing, these bacteria requires highly alkaline media for growth. It is wrong because most of the plaque bacteria which are present in the teeth plaques, they lives in neutral or slightly acidic media, slightly acidic conditions. Okay, and the option C, I mean, third one, R. The bacteria requires a very hard surface, such as that of teeth. That is also correct because these bacteria which present in the plaques, they are sticky. They are sticky to the some surface, so they need some surface. But as we are growing it in the liquid media, it is not able to grow there. That's why there is no growth in the that culture media. So answer is correct answer is D, option D, both P and R. The next question is Bt toxin produced by Bacillus thuringiensis. It does not kill the bacteria itself because the toxin is option A isolated in a special intracellular sac, option B it is an inactive form inside the bacterial cell, option C active only against eukaryotic ribosomes, option D produced in very small quantities. Here the correct answer is option B. It is inactive in form inside the bacterial cell. That is right. Bt toxin, whenever it was present within the bacterial cell, it is inactivated state, and whenever it enters into the insect, so it becomes as a active, and it affects the intestinal cells of the that particular insect. The next question is which of the following is a non-symbiotic nitrogen fixing bacteria? Option A Rhizobium leguminosarum, option B Nitrosomonas nitrosus, option C Astrobacter crococcum, option D Alcaligens fecalis. The correct answer is option C Astrobacter crococcum. Next question is match the pathogenic microorganisms in group 1 with the diseases listed in the group 2. So see here Treponema palladium, Bordetella pertussis, flaviviruses, Leishmania donovani. And in the group 2, whooping cough, yellow fever, kala azar, syphilis. So we already observed the same kind of bit in the previous year also, right? In 2013 also. See here, Treponema palladium causes the syphilis fever, Bordetella pertussis causes the whooping cough, and flavivirus it causes the yellow fever, and Leishmania donovani causes the kala azar. So the correct answer is option B. Okay. So let's move to the next question. A bacillus species divides every 30 minutes. If a culture is inoculated with 1000 cells, how many cells will be generated after 3 hours? Very easy question. Correct answer is option B 64,000. The next question, but last question. The selective media mannitol salt agar is used for the isolation of. Here, mannitol salt agar is a selective medium. It contains 7.5% of NaCl. It is very high alkaline media. It contains high salt that inhibits the most kind of bacteria. Only salt tolerant bacteria will be survived in this culture media. So obviously the option is option C, Staphylococcus. Thank you for watching this video. Please do like, share and subscribe to your friends near and dear. Thank you so much.